This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 224, recorded on March 15th, 2013. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dickson Despommier.
Hello, Vincent.
How are you, Dickson?
Doing very well, Vincent. Thank you very much. And you? How are you doing? No one ever seems to ask Vincent how he's doing.
It's okay.
Okay, Vincent, how are you
I'm
doing?
fine, and you're actually here in my office, not in Fort Lee.
Well, that's true.
Thank you for joining us today.
You're welcome. Pleasure.
Also joining us today from Ann Arbor, Michigan, Kathy Spindler.
Hi, greetings on the Ides of March.
Oh my gosh, it is.
Right.
That's right.
Exactly.
So what happens on the Ides?
Well, if you're Julius Caesar, you better look out.
That's true.
That's it? That's the only thing?
Well,
That's pretty much it.
But the Ides is the 15th, right? That's another word for the 15th?
yes.
Right,
And so he was killed on the 15th.
right.
Now, that's a Shakespearean thing too, right? Because there was a play called Julius Caesar. Okay.
Indeed.
Wow, I learned something. I learned a lot today, Kathy. Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
How are you?
I'm doing okay, but Jan Vincent has a lean and hungry look.
Jan Vincent.
He thinks he's too ambitious.
How is the real line? Is it Jan Yorick? No, that's our last poor Yorick.
Cassius, I think
Different play.
Yeah, it's
Vincent.
different play.
Sorry, I'm not good with that.
Cassius thinks too much.
Ah, yes.
Well, here we are, ready to do another TWiV, and we'll start with follow-up. How's that?
That sounds good.
First follow-up is...
Well, that's what FU stood for.
You're not supposed to say that. First one is from Richard, who writes, the FU humor is tasteless and more apropos for Beavis Butthead mentality, not role model type academics. Love the show.
My goodness.
I like how he just segued love the show down there.
Yep.
I agree.
Right.
I understand. We will do no more tasteless humor.
We will not give the obvious possible response to this letter.
Yeah, right.
Except
No, right. It was. I think it was kind of an inside joke that we didn't really let the listeners in on.
What, the follow-up?
Yeah, because Vincent had abbreviated it. We used Google Docs on the show, and the listeners don't see the Google document that we're working with, but
true.
it's all this shared information for the show. And Vincent had abbreviated it FU, and I had put in brackets underneath it, well, FU too. And then it ended up in part of the audio of the show and
That's right.
became a running gag. But we didn't really clue the listeners into
No.
the gag. So
Do you
not
remember
really
the
fair.
scene from The Odd Couple, the movie?
Well,
When,
there are many scenes, no?
When Felix writes him a note and tells Oscar to clean up his act, and the refrigerator door was left open, you left the water dripping, and the garbage wasn't taken out. And then the bottom of the letter, it's signed F.U. And so
Felix Unger.
It's for Felix Unger, but the other guy thought it was what the other
Yeah.
guy thought it was.
All right, we're sorry
Whatever.
about that.
Yeah,
My
we
apologies.
won't do this
no
anymore.
more. No more.
At least not right away.
No more
Our
lowbrow.
humor is very slightly above that, usually.
Alan, I think you should take the next follow-up.
Sure.
wow.
Jacob writes, Dear TWiV team, I'm a huge fan of your podcasts. I realize a lot of time and energy goes into continuously producing them with such high quality, and I think that is truly outstanding that you do this for us free of charge. I have a comment regarding Alan's influenza vaccine efficacy pick that shows 71.4% vaccine efficacy in preventing hospitalizations. The idea of getting at influenza vaccine efficacy by just screening people who are admitted to the hospital with a suspected respiratory infection is, in my view, somewhat problematic. The idea is that cases and controls will be better matched in terms of risk of disease, acute respiratory hospitalization in this study, and propensity to seek or require care. This should, in theory, reduce some of the confounding bifactors associated with vaccination. This reasoning requires that risk factors for developing a large variety of both viral and bacterial respiratory infections in a wide age span, 18 years and above, are the same. It also requires that the propensity to seek care is similar regardless of diagnosis. To put this in other words, if a patient vaccinated against influenza is admitted to the hospital because of pneumococcal pneumonia, it is hypothesized that he would have sought care if he had acquired influenza. And the fact that he got pneumococcal pneumonia and not influenza is attributed solely to the vaccine effect. In my view, bigger problems with this CID article than the selection of cases and controls is the small sample size. They managed to enroll 169 eligible participants and only 17 had influenza. To draw any conclusions on vaccine efficacy on such a small material seems shaky. I believe the study by Baxter et al., he provides a link, is a much more robust way of getting at influenza vaccine efficacy and preventing hospitalizations. This methodology has its own problems, but in my view is superior to Allen's pick. And he cites this earlier study and then adds someone, Nick, probably has a more intelligent take on this. And I would be delighted to hear what you guys think. Thanks again for a great pod. And Jacob is at the Karolinska University Hospital in Stockholm. And in fact, one of the major reasons that I picked that as a pick of the week is I wanted to kind of continue this discussion that we've had ongoing for a while about measuring flu vaccine efficacy and what do you mean by efficacy and how do you get to that. And yes, this approach is far from perfect. The approach of the study that Jacob just cited is far from perfect, and the approach of all the other studies that have done is far from perfect. I thought the tactic that they took here with what they're calling a case positive, control negative, or vice versa design, a test negative design. Mm-hmm. I thought that was an interesting approach, and I gather a relatively recent innovation that people have started looking at efficacy this way. So I think that aspect of it is important to follow. I absolutely was not saying that this settles the case. Quite the opposite. And in fact, I also contacted Nick, and this is Nick Kelly at CIDRAP, who is immersed in the flu efficacy literature. I contacted him on Twitter and asked for his thoughts, and I've got some links here that we can put in the show notes. There was a previous paper from the same group that did the CID article. They got a somewhat different outcome in an earlier year. And Nick says, this is a series of tweets, so the breaks in it are Twitter's fault, not Nick's. But he says it's similar to their earlier study which we included in our meta-analysis. It's not as clear method-wise this time, but typically it's an okay design. I don't understand how they got a significance over 65 with such few numbers. Also, with their previous paper, none of the 3 years were significant on their own, but with less flu, it was significant this time. Just don't forget it's not a fair comparison to compare this study to recent vaccine efficacy studies in the MMWR, which had different outcomes. And then he also he sent a link to a paper about this general study design, the test negative study design. So for people who'd like to delve into this, I highly encourage following these links and reading these papers and understanding that none of these are probably the correct answer.
The paper that Jacob suggested is interesting because they address this issue where people who get flu shots, in several studies, they found that people who get flu shots get low mortality even when flu is not circulating.
Yes.
Which means that there are other confounding factors, not including flu, and that's the problem with these studies, that you have to get rid of those, right?
Right.
And so they do this difference in difference analysis, right, where you try and compare the effect in when the virus is circulating versus when it's not, to try and get rid of these confounding effects.
Right.
So that's a cool. I'd not really looked at that before.
Yeah, it's a very, very complex, nuanced problem that unfortunately we don't get solid answers out of any of these. But comparing them, you can kind of get into what's going on with this and some of the factors that need to be kept in mind.
This is a thing that happens when you can't just infect people with and without vaccine.
Exactly. We need a randomized, controlled, huge, massive study with millions of people in it, and we can't get that done.
Shame we're not in better contact with North Korea.
Hey, that's what we did for the first polio vaccine,
Maybe
right?
Robert... Dennis
Dickson,
Rodman.
did you know, first polio vaccine?
Yes.
That's what it was, a randomized trial.
Really?
Half the kids got no vaccine, half got Salk vaccine.
Wow.
Well, they didn't know that the Salk vaccine was going to work, so that was
Right.
that was ethically reasonable at the time.
Yeah.
Yeah, we've gone past that point. 'Cause we know we have vaccines for a lot of these. Kathy, could you take the next one, please?
Sure. Justin writes, Salutations, TWiV team. I've been meaning to write this since I listened to TWiV 223 on Monday, but life occasionally gets in the way of my TWiV schedule. I really enjoyed the EVE discussion, which is EEEV, Eastern Equine Encephalitis Virus. And I thoroughly enjoyed the episode title and the viral discussion about snakes. While listening, you referred to the venomous snakes as poisonous. The two terms may seem interchangeable, but for me, I twitched to the level of someone saying that they have stomach flu. Okay, so we better pay attention here.
Yes.
In a poisonous animal, the toxic compound is injected or absorbed, like a poison dart frog, whereas a venomous organism uses a special adaptation to deliver the toxic compound through a stinger or fangs, etc. Thank you all for the wonderful podcasts. Justin.
I didn't know that. Did you guys know that?
Nope.
I
Well,
did not, but
it's-
it makes sense. I mean, I've eaten rattlesnake, and it's not poisonous.
Well, the
Right.
term envenomation has meaning here because the venom is what they're referring to, I guess.
So an animal is venomous if it has an adaptation to deliver the poison,
Right.
but it's poisonous if it's, for example, toxic to eat.
Like the box jellyfish or something like
fugu.
that when you encounter it.
Right. Alan, you should take the next one also.
Yes. So this is follow-up to follow-up. We've had this discussion about protozoa and their retroviruses, which goes back a couple of episodes. Do protozoa have retroviruses? And a friend of mine on Twitter, who goes under Dr. Ricky, had commented that protozoa is, first of all, a very, very nebulous term. But yes, amoebae do have things like retroviruses. And then Vincent's response to that was, well, are they really retroviruses? Or are they just dead transposons? So Ricky says he put a graphic up on a Tumblr blog that he maintains that shows, he says, this is the basal tree for the evolution of all eukaryotes, taken from tolweb.org. Everything labeled with an arrow and also everything in the box falls into the category of protozoa, sometimes protists. This illustrates why the terms are actually morphological descriptors, not evolutionary ones. This diversity, of course, only became apparent in the age of molecular phylogenetics. Even so, the genomic samplings are heavily skewed to a small corner of the tree. So he shows in this evolutionary tree that how bad a term this is, basically. That there's a bunch of stuff that is more closely related to animals, but is in fact called a protozoan under the old terminology. And then on the retrovirus versus retrotransposon question, he says the crew on TWiV are better experts than I, but I think the only real distinction based on sequence between retroviruses and retrotransposons is that the latter have lost the ENV genes that enable them to transpose out of the cell. In fact, one may argue that the presence of retrotransposons in a genome is an important marker of some ancestral retroviral infections, since, of course, the loss of the ENV proteins would select for their retention in the genome. That said, amoebozoan genomes do have families of retrotransposons complete with LTR, polymerase, and high transcriptional activity, and transposition has been observed in a few cases. Is that sufficient proof that protozoa have retroviruses that infect them?
Yeah, it's okay. I just didn't know, so
Yeah.
that's fine. But you have to have these elements to make sure it was an infection and not just some other piece that came in from somewhere else, and he provides that, so that's fine, yeah.
So protozoa, as the term is used, do indeed have retroviruses.
Yeah. Okay. Today we have two papers. One I just found yesterday. It is a letter to Nature, and it's identification of the receptor, the cell receptor for this novel coronavirus-EMC, which we talked about last week, and we have talked about quite a bit recently, because the one that's caused, now 15 infections in the Middle East, and it's called Coronavirus Erasmus Medical Center. The name of this paper is Dipeptidyl Peptidase 4 is a functional receptor for the emerging human coronavirus EMC. Thanks to Steve Goff for sending this along to me. This involves the Fouché group, which is at Erasmus Medical Center, but also individuals in St. Gallen, Switzerland, and Zurich and Saudi Arabia, and Bonn, Germany.
Do you want to do the author list?
Why don't we do the first two authors who are co-first authors, Stalin Raj and Hui Hui Mu, and the senior author is Bart L. Hagmans.
Good.
A lot of names in between.
Yes.
Now, astute listeners may recall that some time ago we did a paper in which from the Fouché group, where they studied the replication of this coronavirus in cells and concluded that the SARS receptor was not the receptor for this virus. I don't know if anyone remembers that, but that's what we did talk about it a little. And they said in that paper, it would be interesting to find the receptor. And now in this paper, they identify it. So consider this virus they've only had since November, I think. They got it from a patient in November, and now they have the receptor. That's pretty neat. It's really fast.
Yeah, and the method that they use, I think, is just really elegant and simple, and it would be a great one to describe to the undergraduate virology class of here's one way to find a receptor.
Yeah, so we know that these coronaviruses, they're envelope viruses. They have a single glycoprotein that is responsible for attaching to cell receptors. So the authors, it's called the S or spike glycoprotein. So the authors expressed the distal fragment of that protein fused to the Fc portion of an antibody molecule. So they made a kind of artificial soluble protein, and this they can first use to stain cells, to add to cells, and then you can detect the presence of the glycoprotein and see which ones bind it, and then later on use it to get the receptor itself. And so in the first one of the first experiments they do is to simply put this on 4 different kinds of cells. Vero, African green monkey, CAS, another monkey cell, HUH, human hepatoma, and bat cells.
And the reason you can do this is that you know that the spike glycoprotein is the binding site.
That's right. Well, for this virus, you don't know, but
you don't. But
the other ones do.
Yeah.
So it's probably since this has a spike, just like the others.
It's a coronavirus, and based on what we know from the basic research on other coronaviruses, we can make a pretty educated guess that that's what's going on.
So you can detect this spike protein binding by immunofluorescence. You can detect it by flow cytometry, and then they correlate the binding of this with how the cells are infected. And so you see this protein can bind to cells that can be infected by the virus. And the Veros and the HUHs seem to produce the most virus, and they seem to have the most binding on their surface by flow cytometry. The bat cells have a bit less binding and make a little less virus. So that gave them some confidence that this reagent could bind cells, and then they try and then use it to pull out the protein. And they mix the protein with a soluble or lysate from HUH7 cells, which are the ones that in their flow cytometry bound the most. And then you can then purify the SFC protein and whatever is bound to it by adding beads that have Cephros beads that have protein A attached to them. Right, Dickson?
Yes.
Protein A will bind the FC.
That is right.
Protein A, of course, is from Staph aureus, which is, its function is to bind antibodies. But it's been adapted to research use for years now.
Right.
And then you can, we say, affinity purify these complexes. Then you run them on a gel and see what's there.
Right.
And, of course, you see the S1FC protein. And then they were so lucky. They had one other protein on the gel.
That's amazing.
I cannot tell you how many times this has been done over the years, and the gels are a mess.
Yes.
This is all true.
You bring down everything. Yeah.
And then you end up going after something, and it's not the receptor because it just happens to be something that was sticking. So these guys are very lucky. So they have this band, they pull it out, they do mass spec, and they know exactly what it is.
Wow.
It's DPP4.
Wow.
Which is phenomenal. That's why it was... Well, see, the thing is, now they have all this there's history and technology available to them, knowledge about other receptors. So you can apply all that, and here's what happens when it works. So this is really beautiful. So they get this protein. It's called DPP4. They identify it by its sequence, and then what you can do a lot of experiments to prove that it is actually the receptor. And one of the big ones, of course, is to express that DPP4 in a cell that normally doesn't bind the virus,
right,
and show that it confers binding and infection.
Yep.
And COS7 are not susceptible, but they are permissive for infection. Everybody remembers those two words, right? Which are properly used in this paper.
Good.
So I think they listened to that TWiV where we criticized them. Yeah, I know they didn't, but I'm glad they have it right here. So the expression of the protein confers susceptibility. The protein itself will bind virus and prevent the DPP. They express it in a soluble form. It will block infection.
right.
They make antibodies to DPP4, and those block infection, right?
Mm-hmm.
And then there's one more thing they did. So we got expression, we got blocking, we got soluble, binding. I think that's it, right? Unless I'm missing something.
But they make the COS cells that express the DPP4?
Yeah, they make it.
Right.
So the soluble blocks, expression makes them susceptible, antibody blocks infection, and then purified protein binds and inhibits infection.
Yes.
Those are sort of like the four main things you want in a paper to really prove it's a receptor, and they got them all.
Mm-hmm.
Nice.
I got to tell you, when we did the polio receptor in 1989, we did not have expression of soluble receptor and binding to virus. But they let us publish it anyway, which was nice of them.
Well, that was back in 1989.
It's ancient history, right?
Yeah.
Yes, it was one of the first.
Have you
viruses.
subsequently done those two
Yeah, we did.
missing experiments?
Oh,
Did
absolutely.
They prove out okay?
Yeah, we made a soluble form of the polio receptor. It blocks polio,
Right.
and it binds to the virus. It blocks infection. And then we selected virus mutants that overcame the block and to identify the binding site. It was really cool. And I
Nice.
presume that's what these guys can do as well.
Mm-hmm.
So this protein from bats, they have a bat version of it. It also confers susceptibility, but just not as well as the human.
Right.
So they are saying maybe, when this virus, by the way, is believed to have come from a bat, because there are related viruses known to be in bats. So maybe the virus evolved a bit to infect people, right? To bind better to DPP4. So if they can get the bat version that was the precursor of this, they could test that.
And the DPP4 protein is very highly conserved.
Yeah.
So it's pretty similar across different species, but apparently it's different enough between bat and human to provide some sort of some slight
Right.
difference in infection.
Now, DPP4 is interesting because, as the name says, it's a peptidase. It's actually an enzyme, but it's in the membrane. Right? That's unusual. But there are others like that. In fact, some other coronavirus receptors are similarly enzymes, peptidases in the membrane.
Right.
And so what this one does, I think, is to is to cleave cytokines and regulate their activity. And apparently this DPP4, in addition to being membrane-bound, is also found soluble in the serum. So maybe, there it has some role in regulating cytokine activity. So they wonder if this has something to do with the pathogenesis of this coronavirus infection.
Right, but they did find that inhibiting the peptidase activity of the protein doesn't seem to affect the virus infection.
Yes, that's right. And that's true for the other coronas as well,
Right.
that you don't seem to need peptidase to infect cells.
So it's not like a flu type of interaction or something where you need a cleavage going on.
Which would have been cool, right?
Yes, would have been very cool.
To bind a receptor that then
Right.
cleaves your glycoprotein so it fuses.
Yeah.
That would be very cool.
So have they ever actually identified the source of the original SARS outbreak in China?
Well, yes, they do have what they think is the precursor virus, yes. And they can tell you how many changes it underwent to get in people, yeah.
I mean, there were a lot of changes in the beginning, I recall. That is, they couldn't find a close relative to begin
Yeah,
with.
they've got what they think
So
is
they
the
have it now.
precursor.
Is that in a bat also?
Yeah, it's from a bat, and I think it went through a civet.
Oh.
And then went. So the bats somehow got the virus into civets, which were then brought into the open market and infected people.
Okay.
So this DPP4 in people is found on the epithelial cells in the respiratory tract, which makes sense, right?
Yep.
Because that's where these people are being infected. It's also found on the kidney, small intestine, liver, and prostate, and on leukocytes. So I think some of these individuals, they isolated virus in their urine. So maybe it's somehow getting to kidney and replicating. And that wouldn't be unprecedented for a respiratory virus. Measles, gets in by the respiratory route and then goes into the blood. So it'd be interesting to find that out.
Yep.
So having the receptor lets you start addressing all these questions. Where is the virus going?
Nice.
It's also interesting that it's found on non-ciliated cells, whereas the ACE2 receptor for SARS is on the ciliated cells.
Right. They called that, what was the name of those? It's something to do with tubulin. Where is that? Non-ciliated. Yes.
Tubulin for
Negative.
minus, because I think the tubulin must be part of the cilia. That was my reading of it.
And so if you remember from the last week, we discussed primary human respiratory cell cultures. So they express this receptor, and they are, of course, susceptible to infection as well.
Right.
And they have some tissues here, too, some human tissues which they stain, and they show the cells that are infected or the ones that express this receptor. I guess they're all primary cultures. They're not tissues, right?
Right.
I don't see any tissues here. Yeah, they're all primary cultures. So it would be interesting to do some sections of intact organs and see where this is in the respiratory tract also.
Yeah. Now, this could also be helpful with looking at animal models.
Right. Now you can see what animals have a close version of this, and that might be a good model, right?
Yeah.
Mm-hmm.
Yeah, I'd look in mice right away because that would be convenient, right?
Mm-hmm.
Sure.
It'd be terrible if you had to use ferrets.
Well, they're bigger and more expensive for one thing.
Right.
Or bats.
And they're not humans.
Right.
Bats would be difficult. Yeah. Although there's some labs in the world that do bat grow bats. They have colonies and they infect them with viruses.
You have to do all your work at night, though.
This is true.
Let's see. Is there anything else curious about this?
They go a little bit into this idea that this could. You touched on it a moment ago about the potential involvement of DPP4 in pathogenesis.
Yeah.
Where affecting the level of this could affect your ability to process your cytokines, which could affect your immune response and feedback.
So they cite this ACE2, which is the SARS coronavirus receptor.
Right.
They say it's been shown to be downregulated during infection, and that contributes to the severity of disease. Yeah, so that could be for here as well. But so they'll have to check that out.
I just happen to have one of the tables from the supplement up on my screen at the moment, and I noticed that they didn't find any surface binding of the EMC virus to one cell line that they looked at from mice. So that might mean that they'd have to make a mouse.
It could be a problem. Yeah, they could make it transgenic, right?
Yeah.
That would be okay.
Yeah.
Well, in fact, that could actually be advantageous in some ways because you could maybe muck around with what tissues express it and see if that's an important determinant of tissue tropism.
Yeah.
Mm-hmm. And it is only one mouse cell line that they list in this table, so they might have picked the right one. I was thinking, Vincent, when you pointed out that they got this single band, that even if they had gotten several things identified in their mass spec, they would have had the information that the the receptors in the other coronaviruses are these peptidases, and they might have gotten this as a hit and then gone after that.
Yeah, so that's one way that it's different now. I mean, 20 years ago, if you got a couple bands, wouldn't be easy to figure out what they were. We didn't have the widespread availability of mass spec. It would be really hard to sort out. I know examples of people who purified proteins, candidate receptors in this way, and they were not the virus receptor.
Right.
You know? But today you got mass spec. You know, if you got a couple of bands, you could sequence them all. It's like $200 per band. It's not that expensive.
It's quite amazing.
And then, well, it's like the DNA sensor paper where they had a couple of candidates and they picked one out that looked good and followed it, right?
Yeah.
So you can do that.
Yeah, and in fact, if the first one that you pick doesn't work, you can follow 6 or 8 leads, and that's not too big a deal either.
As long as you have people who can do it.
Yeah, but I mean, some of the preliminary experiments you could knock out of the way reasonably easily. You could see, all right, we've got this lead, let's express it, and let's see if a soluble form inhibits the virus.
Yeah, we had Terry Dermody here this week, and he put this in a good way. You know, you identify a putative receptor, then you find someone who works on it, and you ask them for for their freezer.
Yes.
Can you send us your freezer contents? And sometimes people are nice enough to do that. So are you going to do this with your class, Kathy?
Well, we're already past talking about receptors, but I was just thinking that for a visual for a class next year, this might be really easy to do.
It's a clean one. It worked. It's a good story because it's clean and straightforward, yeah. So you see how this virus has killed less than 15 people, yet lots of people are working on it. And I know that... forgot his name again. I forgot.
I think you want to say Matt Freeman.
Why am I so bad with names? Am I like the guy who can't remember faces?
You mean Oliver Sacks? No,
Matt Freeman is working on it.
You're not.
And he said he wants this to be his SARS. So I don't want it really to spread and kill people.
No.
But
no.
It may be biologically interesting anyway to work on. So what a cool story, right?
Yeah.
And there are a couple of things in the front part of Nature, the same issue. So there's a nice News and Views by Tom Gallagher and Stanley Perlman about coronavirus receptors, and then another one by Declan Butler. Great. So you could lots of ways to read about it, and it's an easy paper to read it in itself. So,
Right.
All right, our second paper is in Science, and the way I came across this, which often happens in science because the papers run into one another. One of my colleagues on TWiV had sent me the previous paper. Was all about the difference in the gut microbiome between men and women, I think. And I was scrolling through it, and I came upon the beginning of the next article. So in science, that's how they work. And this one, I said, oh my gosh, this is really interesting. Interferon-epsilon protects the female reproductive tract from viral and bacterial infection. So I'd never heard of interferon-epsilon. So I read the abstract, and I got hooked. Dickson, you ever get hooked by a paper?
Absolutely.
And I'm happy when I get hooked because it means I'm still alive.
Yeah, that's right. That's right.
Scientifically alive. Because if you don't get hooked, after a while, you're just, it's bad. It's really bad. Yep.
So, Vincent, the same thing happened to me with the end of this paper because it reads into a paper about migraine headaches. And so I read that abstract.
Oh, yeah.
So
with that, I don't understand at all. Did you understand?
No, but I just,
so that's a feature of science that they, one article ends and then immediately, just to save space, I suppose, they start the next one. Where in other journals, there's...
Yeah, that's right.
And they keep them separate, and you have less chance of serendipitously finding a new topic. Anyway, as everyone knows, and we've talked about here on TWiV a lot, interferons are part of the innate defense against microbial infections, and they operate very early on when you're invaded.
Right.
And we have talked about type 1 interferons, alpha, beta, and omega. So omega is a new one to me. I know alpha and beta. And what happens is that when you're infected, the cells sense infection, and we talked about DNA sensors some time ago, and then they start a signaling pathway that leads to the production of interferons. Now, in this paper, they say within the locus that encodes the type 1 interferons, the genes for type A, B, and omega, there was a gene which was called interferon-epsilon, but nobody studied it. So
Nobody knew what it did.
Nobody knew what it did, so they decided to study it. And one of the first things they do, which is the type 1 interferon signal through receptors on the cell surface. These are called interferon IFNAR receptors, IFNAR. And so what they do is to ask if you take away these receptors, does it make any difference to what interferon-epsilon does? So they add interferon-epsilon to macrophages, which are made from bone marrow of mice, and they look for the induction of typical interferon-stimulated genes, ISGs. They look for two, IRF7 and 2' 5' oligoadenylate synthetase. And these get induced when they add epsilon, but if you use macrophages from mice that lack the IFNAR receptors, there's no induction. So they say
it's a legitimate interferon.
Legitimate type 1 interferon,
Right.
which I guess makes sense because it's in the gene cluster, right?
Mm-hmm.
Yeah.
Then they ask if you stimulate some of the known sensors, like toll-like receptors and DNA sensors, does this gene get induced? Because this is a typical effect of stimulating these sensors. The other interferons are induced in a feedback loop.
Right.
So they don't see any stimulation of interferon-epsilon when they tickle these sensors, these toll-like receptors and DNA sensors with all the right ligands, which is kind of interesting, right? So that means that there's no this interferon-epsilon doesn't respond to known sensors.
Right. So it's an interferon, but it doesn't seem to get turned on like one when there are pathogen-like things around.
Right. So this, of course, is being done in cells from mice. And so the next thing they can do is try infecting these cells and see if there's any change in interferon-epsilon expression, because with type 1 interferons, alpha and beta, they go up when you infect cells with various microbes. They use some leaky forest virus, which they showed induces 2' 5' OAS, but not interferon-epsilon, and neither does herpes simplex type 2 or chlamydia.
Right.
They don't induce interferon-epsilon. You okay so far, Dickson?
Absolutely. I'm following this with avidity.
Avidity?
That's right. And claridity.
So here we are. This is a type 1 interferon, but it's not regulated in the normal ways that it certainly signals through the receptors, but it doesn't seem to respond to the usual sensors. So what's going on? So they said maybe it's just expressed all the time. And that in most organs they looked at in mice, it's not detectable, but they found it in uterus, cervix, vagina, and ovary of the mouse, and particularly in the endometrium. So interesting. Constitutive expression of an interferon. It's quite interesting.
Yeah.
Mm-hmm.
They also show that the expression varies about 30-fold during the different stages of estrus. So during pregnancy, it's way down. So they said maybe it's hormonally regulated. And in fact, if they take mice where they remove the ovaries and then give them sex steroid hormones, they show that it induces the expression of interferon-epsilon. So it's hormonally regulated, which is something totally new for an interferon, right?
Mm-hmm.
Yeah, I think so.
Mm-hmm. I think so too.
And then they looked in human tissues, and they find very little expression in most tissues, with the exception of endometrium. And in fact, they have cells, epithelial cells from that site from 6 different women, and in various stages of menstrual cycle or after menopause, and they find that the epsilon expression is the highest when estrogen levels are high.
The mice weren't lying.
The mice were not lying. Exactly. It's nice that they went ahead to do this, because Dickson asked me this morning when I told him about this. He says, well, what about in people?
Yep.
Right.
There you go, Dickson.
Or what about the same cells producing two different kinds of interferon. One inducible and one constitutive.
Are you asking whether these cells produce alpha and beta?
I am.
I don't remember the answer. Does anyone recall whether they looked at alpha and beta?
I don't recall.
I'm wondering if in the maybe in the supplementals, did they look at some IFNAR deficient? I don't know. I can't remember either.
Yeah.
But what I wanted to say is that what they looked at in the mice, which have an estrous cycle and not a menstrual cycle, but the fact that this is the highest in the stage called estrus is when the female is sexually receptive, makes sense from a biological standpoint, because that's when you're most likely going to be having foreign organisms coming in, and you'd want to increase the level of this interferon.
Yeah,
I thought that was really cool.
Yeah. And
and then it declines in pregnancy during implantation because you don't want to reject that.
No.
Yeah, and you don't want inflammation caused by interferons being up and all that stuff. Yeah, it makes sense. And this is cool. This is totally new, right?
Yep.
I mean, this makes total sense. Then they test infections in these animals. They make actually knockout mice for the gene. So this is a big deal. That's a lot of work. Interferon-epsilon knockout mice. And they infect them with either herpes simplex type 2 in the genital tract, which of course is a cause of STDs, sexually transmitted diseases, and get more virus produced without epsilon. They get more clinical severity and more lesions compared to wild type.
They didn't really need to include a photo of the mice with herpes, but they did. It's in there.
That's right.
Yeah.
And they say, though, that in these mice you can protect them by giving them interferon-epsilon.
Yeah.
Which is nice to show that is actually, by taking away the gene, that's actually what you're doing to make the infection more severe. And there's more, I think they find more virus for simplex 2. They find more virus circulating, right? What is that? No significant change. No, that's the wrong one. Yes. Higher titers in the spinal cord and brain 7 days post-infection. So, by not limiting infection in the genital tract, then it's more likely to spread to the brain and spinal cord.
Right.
Right. I wanted to point out that because of the fact that there's these varying levels during the estrus cycle, it occurred to me that, well, how do they know that they're not just infecting their mice at different times, but in fact, they sort of synchronize their mice before infection by giving them the Depo-Provera, the progesterone.
Right.
So that I think that's their way of making sure that's not a confounding factor.
Yeah, good point. That's good, yeah. Yeah, that would be important because you wouldn't know, right? By looking at them. So you synchronize them to make sure. Yeah, that's good. They also use chlamydia, which is another sexually transmitted organism, bacterium, of course, and it's apparently the most prevalent bacterial STI. And just like with herpes simplex type 2 in the epsilon knockout mice, more severe disease and more bacteria in the genital tract. And do they do an epsilon treatment here? Yes, you see a dose-dependent reduction in bacteria when they administer interferon-epsilon to the infected mice.
Right. One thing that, back on the herpes part of it, they said, notably there was no significant change in the expression of interferon-epsilon in the first 3 days after viral infection, consistent with our in vitro data showing that this gene is not pathogen-induced. And that's a supplemental figure.
Right.
And while they don't see an increase, and I think they don't have enough mice to have it be significant, as the infection goes on, they see lower relative levels of this. And so it wouldn't surprise me if the virus has some way of reducing the interferon-epsilon.
Yeah, right.
That's what it said to me, but they need more numbers to make it statistically significant.
Right, so that's right. That's a good point. So the key here is that this epsilon doesn't respond to pathogens, right?
Right.
It's constitutively on and is effective, but the virus is probably overcoming it. Yeah. I'm sure people are going to look at that because that's very interesting.
Yeah.
All right. So the idea here is that now we have an interferon specific to the genital tract. Females, right? They didn't find it in males. And this could protect, as Kathy said, during mating. And in people, maybe, STIs are a big deal, right, globally. Mm-hmm. And maybe this is a way to treat them or somehow interfere, right?
Mm-hmm.
Yeah.
Sure. There is one wildlife species that I'm sure the listeners are aware of that's becoming reduced in numbers, not to extinction, but certainly to critically low numbers, and that's koala bears. And their sterility is being caused by a chlamydial infection.
Yes.
And I'm just wondering whether or not this kind of data could be used to create a wider stimulation of the interferon-epsilon in that group of animals.
Well, it turns out that these interferon-epsilons are conserved in eutherian mammals.
Yep.
Right.
But they don't talk about marsupials.
Right. So that would be interesting to know.
Mm-hmm.
So, Dickson, those koalas are getting chlamydial infections,
Yes.
it is thought, because they're infected with a retrovirus that is immunosuppressive.
Yes.
The koala retrovirus. And
Okay.
they're immunosuppressed to a certain extent.
We talked about this, didn't we?
Yeah,
we did.
I vaguely remember that, and just wondered how you would connect that up with this data, that's all.
So what is a eutherian?
Well, some definitions say that it's a placental, but it turns out that there were some non-placental eutherians, but they're
Right.
extinct.
Okay.
Right.
Okay, so right now on the Earth, the eutherians carry a placenta.
In practical terms, it now means placental.
So
Right.
this would make sense because it's protecting the
Yes.
area, as you suggested earlier, Kathy, right?
Mm-hmm.
So because, of course, you have to have fertilization and live birth, so you need this epsilon there.
So you wouldn't have to worry about monotremes then.
Sorry, you wouldn't have to worry about
Monotremes. No, they're in a whole different department.
what's a monotreme.
It's an egg-laying mammal.
Okay. I was just going to say, I'm asking you guys for definitions because I'm not going to get into that.
Right. The eutherians are placental mammals nowadays. There were some non-placental ones earlier, but they're gone. The marsupials are the other
Yes.
major branch, and then the
Yes.
monotremes are the third branch that is only represented by the platypus and the echidna now?
Yes.
So these are egg-laying mammals?
Yes.
Yes.
So did you know that there is a theory that the reason why eutherian mammals developed is because their precursors were infected with a retrovirus that provided the syncytian protein to allow fusion of syncytial trophoblasts in the formation of a placenta. And syncytian is related to the envelope glycoprotein of retroviruses. And in mice, so it's from a retrovirus, in the endogenous retrovirus. And in mice, if you knock it out, they don't make placentas. We have those genes and we express them. And it's thought that it's why we can make placentas. Can you imagine an infection? A viral infection made egg layers become those with placentas.
Well, maybe it was because of the particular course load I was taking at the time. I took parasitology and then embryology almost contemporaneously. And I remember in embryology learning about the implantation of the placenta and how all this develops. And I thought, my gosh, this is an infection.
Exactly.
It's totally an infection.
Yeah,
it invades the wall and it does all this. It's just complete
That's right.
infectious. Life is a sexually transmitted disease.
Here, here.
I was talking to Kurt Anderson on Studio 360 a couple weeks ago, right?
Yeah.
And I was trying to convince him that many viruses are good. So he said, give me an example. And I gave him the placenta syncytian thing. And he cut it out. He didn't read the episode.
Oh, man.
I mean, don't we want to tell people that microbes are good, right?
Absolutely.
Yeah.
But the news is not interested in that. They're only he was interested in zombies, really.
Hey, zombies, that sells these days.
It does sell.
That's right. That's right.
Everybody's into zombies.
But don't you think that the fact that a retrovirus gave rise to a placenta, isn't that so cool?
Yeah.
Well, you guys are all sold.
So-
I'm
yeah, it's
speaking
an
to
amazing.
Oh, yeah.
Preaching to the choir.
preaching to the choir.
Viruses are us.
Yes.
There you
right.
go. There you go.
Did we use that as a title? I think we did.
I think we
I think so.
In fact, they are us.
All right, let's do some email.
Before we do, I meant to mention back when we were on the coronavirus receptor paper that in today's issue of Science, they have several stories that are about the 10th anniversary of SARS. And so those of you that might be interested in that, it's a nice history and a timeline that goes across the multi-page article and tells a little of the, the case studies and the political ramifications of finding out that this is a disease in China and so forth. So take a look
cool.
at that.
Yes, that's very nice.
And that really, I think these articles do a nice job exploring this. It was really kind of a watershed moment in modern public health, I think.
Yeah.
Mhm.
Yes.
It changed a lot of the discussions that we have about emerging infectious diseases.
Boy, I'll say.
Yeah, for example, they'd had some agreement from 1969 called the International Health Regulations, and they spelled out how countries should report and handle diseases, but it only covered plague, cholera, and yellow fever.
Yes.
So something was revised in 2005 and took effect in 2007. So that, yeah, is important.
There's a nice introduction by Martin Enserink, and his last sentence is great. Although SARS painfully demonstrated that sweeping an epidemic under the rug is counterproductive, few people believe that this age-old reflex won't occur again.
Yes.
Wow. You know what he's talking about, Dickson, right?
Yeah.
Do you?
Yeah.
I'm not convinced.
AIDS.
No. SARS first appeared in China.
Right.
And,
No, I went to China the year afterwards to help
They
with
tried
their...
to put it under the rug.
well, they lost $60 billion in tourist trade.
Hong
That's
Kong, it'll
why.
take your breath away.
Arg.
It's a famous poster that Ian
Arg.
Lipkin always shows.
Arg.
It's a travel poster, Dickson. Okay, I'm going to read the email from Jacob. Hi, everyone. I'm rushing to write this email before you start recording the next episode because I just finished listening to last week's podcast and I wanted to get some follow-up comments, questions in. I'm sorry you missed the deadline, Jacob, but you're not too late. Quick background.
Cruel.
No, I'm not being cruel. I mean, I got it the day after we recorded this episode, so. But here it is. I usually try to avoid emotionally charged discussions on social media, but recently I saw a post from an acquaintance that scorned flu shots. My response led to a Facebook debate. See attached screenshots edited for privacy of the others involved. I didn't get the screenshots, I'm sorry. In which I was horribly outnumbered and ultimately left me feeling pretty hopeless about changing public perceptions of vaccines. When I started listening to TWiV 217, I was really excited and hoped that there would be some additional material for me to make my case to the layperson. A good portion of your discussion, i.e. the increased cases of narcolepsy post-vaccination, would only be further misunderstood if I sent them a link to the podcast. While you guys eventually did make sure to say that vaccines are a good thing, you did it in such a way as if it was obvious to you and didn't offer much of an argument for that point. Unfortunately, it seems like there are many misled people out there for whom these statements are much less obvious and apparently even seem like part of a conspiracy theory. Again, see attached screenshots. So my question is this. How do you go about trying to convince people who are uneducated about viruses and vaccines or who have been victims of misinformation and sensationalist media about the importance of vaccination, especially in the case of flu shots, where the rate of protection is not as high as with other diseases? Do you even bother? I was honestly really hoping there would be some sort of video pick of the week that is short and convincing that I could share, because I feel that would be the most effective medium for convincing the average social media user. Thanks for your help and for all your podcasts. I really enjoy listening to them while working in the lab. I'm a fifth-year PhD student in chemical engineering at RPI in Troy, New York, studying the design of inhibitors for HIV and influenza viruses.
Wow.
Well, my philosophy is, yes, you're right. You're going to run up against individuals who will not listen to us, but you just keep hammering away. You just do it over and over again. Every week you reach a few, and you keep doing that. And if enough people do it, eventually you make some headway. But there are some who are very recalcitrant. And in fact, even on the TWiV page over on Facebook, there are people who just will blanketly just deny everything we say. We're wrong. We don't know what we're talking about. You just can't let it bug you. And when it gets personal, you just shut off the conversation, of course. But you have to just keep going back to the science and emphasizing it. Now, I know we get a little complicated, so we don't have the bite-size bits to hand over to the public. What do you guys think?
Yeah, I would agree with you, Vince. I think the press doesn't want to burden the public with extreme detail like we go into. So it's very difficult to convey a complex idea simply.
So you give up, Dickson?
No, not at all.
What do you think, Alan?
Well, I've kind of evolved in my approach to this sort of thing. It does come up a lot in social conversations, but especially on social media where people are hidden behind a keyboard and they're willing to be more blunt and forceful in arguing points they might not believe quite that strongly. But it turns out that a lot of the social science research on this suggests that it's fine to talk about the data and talk about the science, and probably a really good thing to be as transparent as possible about that. But ultimately, anecdote does have a lot of power, and so people tell these stories about, well, I heard a friend of a relative of a so and so say that such and such happened after they got a flu shot, and that actually sticks in people's minds emotionally. So the answer for those types of anecdotes is probably other anecdotes. And in that spirit, I think I picked this many episodes ago. There's an organization called Families Fighting Flu.
Right.
That I wholeheartedly support, but hope never to qualify for membership in. They are made up of parents of children who died of influenza. And needless to say, they are outspoken advocates of influenza vaccination. So I've provided another link to a more recent video from them. They've done ad campaigns with this same general theme. I find it very difficult to watch their videos because they're emotionally wrenching if you're a parent, but that's the whole point. They introduce you to a child and show you what this kid was into, and then he died of influenza and was not vaccinated. Now, as scientists, we look at that and say, well, that's N equals 1, But honestly, that carries more weight in some of these types of conversations with people who don't appreciate the meaning of N. Right. And that is another tack to take when you find yourself drawn into those debates. So there's a video link for you. It's a YouTube video you can point people to and say, hey, you want stories about flu vaccination, here's some more to throw into the mix.
What do you think, Kathy?
Well, I haven't looked at this video, and when you were talking about anecdotes, I'm looking around my office. I found some wood to knock on, because I get the flu vaccine every year, and I have not had the flu that I know of. In my adult life, I don't know if I did as a kid. So, yeah. So, but we all know plenty of people that don't ever get the vaccine.
Yeah. Correct.
So,
and actually, that's another issue that has come up in some of the social science research that I've seen on this. That there's some studies show that a doctor's or a scientist's own personal decisions carry a lot of weight in this conversation. So people are not nearly as swayed by their doctor saying, there's good clinical evidence for the efficacy of flu vaccine in your age group, yada, 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 and just delivering the standard spiel. It can be much more persuasive for the doctor to say, I got mine.
Yeah.
My child got his.
That's right.
All three of our kids have had every vaccine, from the beginning, and they get a flu vaccine every year. They get flu mist, and they've been healthy.
Maybe the best way to do this is to have a Saturday morning cartoon show about this and call it the Flu Fighters.
I gave a guest lecture in Stuart Firestein's ignorance course this week, and this was one of the questions. So one of the students asked, how do you deal with the anti-vax as a scientist? What do you do? It's the exact same question. People want to know what to do.
Yep.
I said the same thing.
Yep.
Keep hammering at it.
yeah.
Okay. Hey, Dickson, are you up for reading a letter?
You betcha.
Justin.
I will read Justin's letter. Justin writes, Hello to the TWiV team and guests. I am wading my way through the long and wonderful TWiV archives since I didn't start listening until about TWiV 170. And I have a question not directly related to a specific episode, but just a curiosity that arose. During the learning period of the immune system, I wondered if it's possible to expose the developing organism to antigens to a pathogen that would cause the mouse or other organism to recognize the pathogen as self and allow for an infection with little or no immune response. Would there still be innate immunity, and to what extent would the immune system be able to respond? I know this is more of an immunological question, but I'm curious on your thoughts. On two side notes, I first wanted to share everyone's sentiments and thank you for all your wonderful podcasts. And I have a listener's pick. I don't know if this has already been chosen, but there is a great store at, and then he lists the website. I personally have the playing cards. Let's see, I'll read that again. I personally have the playing cards, had the temporary tattoo, and the helix tie. Enjoy. So, in answer to the first part of the question, well, I learned that you can tolerize animals to various soluble antigens and to, I'm not sure how much beyond that you can go if you took an entire organism like a protozoan or a helminth and gave it to a developing fetus, whether the organism would then be included into the immune repertoire of the animal and say that's self rather than non-self. Does anybody have a
Yeah, I think
better answer?
There are models where you give viruses shortly after birth where the immune system is poorly developed, and then they are tolerized, and the animals remain infected and they're healthy throughout life until you make an immune response. So LCMV is like that. You can infect young mice and they live, and then if you give them virus-specific, say, T cells, then they die.
Suggesting that the innate immune response.
Well, it's adaptive because innate is not gonna be tolerized to these things.
Yeah, that's what I was going to say. Yeah. You can differentiate between those two things.
Yeah, for sure. I don't think it's a good therapeutic approach.
No.
But it's been done experimentally.
Right.
Yeah, but it shows Justin's really thinking.
Yes,
good.
Very good.
Yeah, and you might. Yes, that's true. Keep it up, Justin.
This is the DNA store he sent us a link to.
I like the bumper sticker, I stopped for UAA.
That's great.
Even I got that one.
There's some cool stuff here.
My car actually looks a little like a ribosome, so that might be appropriate.
Yeah, it's very good. There's some good stuff.
That's very
nice. Funny. There's some very geeky stuff, too, but it's good. It's all good.
Geek is good.
Nice. It just goes on. It's a long page. Thank you,
is,
Justin. Stephen is sending us a link to a Nature article, When Google Got Flu Wrong.
Uh-oh.
Ah, yes.
Do you know what this is about, Alan?
Yeah. Google 2 years ago or so launched a site called Google Flu Trends. As everybody knows, Google's business model is to mine as much data about everybody as possible in order to sell it to advertisers.
Yeah, right.
And seriously, that's the whole concept of Google, is to figure out what you're into so they can sell it to you. But they also, being a huge company, they wanted to do some good, and they thought, well, we're mining all these data. Maybe we can make predictions about how bad the flu season is. And so they started mapping search terms. There are data showing that people do more internet searches related to illness when they're ill, and you can start to map this down. And so Google came out with this tool called Google Flu Trends that tracks where flu is based on people's search terms. And it had some success. It had some success in past years, and this year it didn't work very well at all.
Right. Mm-hmm. Nothing beats virus isolation.
Right.
So you may remember not too long ago we had a discussion of this season's severity, this season's flu severity, right? And the press was making a big deal of it. And a reporter for USA Today called me and he said, this Google Flu Trends is saying it's going to be really bad. And I said, yeah, well, if you look at the CDC virus isolation, it's not, and that's what you should look at. Uh-huh. And it turned out to be not a big deal. So, and I object to this, to the headline on this figure in the Nature article, the latest U.S. flu season is more severe and has caused more deaths than usual. Well, this year was higher than last year, but if you just go back, you see that there are other years that were worse. So usual is not really the right word. Mm-hmm. Come on, Nature. You're a science publication. You're not USA Today.
Yeah, but this actually, it's an interesting news item that highlights a problem with these types of search-based, and people have tried doing social media-based tracking of things. Yes. There's a feedback loop in these systems.
Right.
And so if there are a lot of news stories about how bad the flu season is, there are going to be a lot of search terms about how bad the flu season is, and that is going to skew your results because that's what you're tracking.
Yeah.
It's a very difficult line because then you have to try and figure out, well, how much of this is from all the noise and how much of this is actually signal.
We had I did a TWiV at ICAC last year, and we had what's his name? John Brownstein, who is a Harvard epidemiologist who has been involved with these health maps initiatives, the interface between software and health info. And if anyone, I'll put a link to that because we had a good discussion. I mean, I asked him, can people game it? Of course, they could game it. They could just call up and, say you got, you have a flu remedy or something. So, yeah, and I said, the best thing is still a virus isolation by CDC.
Right.
Okay, there's the link. Kathy, can you take the next one?
I went off the page. Hello, professors. Here's to say thank you for your wonderful podcasts and letting you know that you have a fan from North Island in New Zealand, too. I'm a Japanese female now living in New Zealand, married and a mother of 2 kids. I have no academic background on science, but I have always loved to read about it. I love reading, loved adventure and fantasy stories when I was a kid. When I discovered science fiction, I totally immersed myself in that fascinating world. My primary motive to study English was so that I could read science fiction in the author's own language. Although Japanese translators are doing a marvelous job, and nothing wrong with reading them in Japanese. When I discovered podcasts, I immediately fell in love with them too. One of my first subscriptions on the iTunes was Futures in Biotech. The episode where Professor Racaniello talked about flu with Mark was really informative and made me feel somewhat in control. Being in the southern hemisphere, we were expecting that awful flu in the coming winter, meaning very soon from the time of that podcast. Before I listened to the Futures in Biotech episode, I received a flyer from the government, How to Prepare for Flu Pandemic. It warned us that shops and public services may not be able to operate if too many people get sick, and advised us to stock up with food, water, and batteries. Also, it encouraged us to set up communication and support network to arrange somebody else to look after your children if you get sick. All in all, everything sounded so grim it made me very nervous. But after I listened to the podcast and did everything recommended there, I had peace on my mind. Luckily, the flu was nowhere near as bad as expected, and TWiV was added to my subscription. In one of the TWiV episodes, it was mentioned that there's no definitive preventive measure for flu, but I'd like to share my experience. When I was pregnant with my first son, meningitis broke out in New Zealand. Many children and young students fell victim to the disease. The bacteria, the main cause of the disease, were said to infect orally, and they said children and students were infected through not-so-clean cups, plates, etc. So, again, hand washing was the best and almost the only preventive measure that we could take. Then one of my colleagues fell sick, was hospitalized, fell into a coma, and passed away. It all happened within a week. She was a senior living alone and nowhere near where infected cups were. I called my midwife, but the only advice she could give me was to visit a doctor if I was in doubt. So again, I did the only thing I could think of, washed everything before putting it into my mouth. As well as washing my hands, I washed cups, plates, and cutlery, anything that I needed to touch from the lunchroom before eating. I washed my hands and face thoroughly when I came home. My son was born healthy to everyone's relief, and I followed this routine faithfully after he was born. I took maternity leave for about a year, then went back to work. When I started working again, I got a little slack with my washing routine, and I got the flu. At that time, I realized that I hadn't had the flu for over a year. Before that, I used to catch cold and flu 4 times or more throughout the year, and I thought that was just my weak immune system. I went back to my washing everything before I eat routine, and I've been successful by not getting sick so often since then. My sick leaves are consumed by nursing my kids when they catch cold. So I am the proof of how effective hand washing is to prevent yourself from flu. Just a little request, please. So just a side comment. So there we have an N of one anecdotal thing. But, there's nothing wrong with washing your hands too often. So very often. So that's good. Just a little request, please. I am now so interested in biology and I want to study. I tried Biology Demystified. It was fun reading, but virus was covered in only one page. But the series is great for an amateur like me. I feel I should try chemistry and biochemistry before tackling microbiology. While I'm making slow progress on these, would there be a chance of Virology Demystified coming out? Or if you have any recommendations of a self-study book on virology, about 300 pages, hopefully, for non-academic, totally amateur people like me, please let me know. Thank you again for sharing your knowledge in these fun podcasts. Yes, I now listen to all 3 podcasts of yours, and I'm fascinated by the unseen world of microbes. Live long and prosper, small and big together. Signing off, Nico. P.S. Wouldn't it be wonderful if we could get flu vaccines with this? If you could buy these over the chemist counter or even have them delivered to your door. And she puts a link into an injection mechanism that I think we've seen before, or at least I've seen pictures of it before.
Yes.
Yeah, the microneedle patches,
Right.
Put it on with a Band-Aid. They're very cool. They'll be here soon.
Well, and in fact, there's a small needle injection system for the current flu vaccine, I think. It's not
That's right,
quite
there
this,
is.
but it's,
Yes, it gives you a smaller dose, saves the dose. Yeah, that's right. One of the companies makes that, right. Well, I would recommend Carl Zimmer's Planet of Viruses, I think, if you want something really simple. A number of people say it's good. Have you guys read that?
I have not, but I have a recommendation that's only about less than 150 pages by Lauren Somperak.
Oh, that's
How
right,
Pathogenic Viruses Think, Making Sense of Virology. It's a very conversational book. Yeah, we've had this on TWiV before.
Yeah, that's right. That's a good one, too.
Yeah, and Zimmer's book is good.
Maybe I'll put links to those.
The only drawback to Planet of Viruses is it's specific viruses in specific settings.
Right.
But by teaching by example, I think he covers a lot of good material.
Right, and that's kind of the philosophy in the how pathogenic viruses think. So there's influenza, rhino, measles, rota, adeno, hepatitis. Well, it goes quite a hepatitis A, B, and C. HTLV1, HIV, herpes simplex, papilloma, emerging viruses. So by the time you cover all those, you've hit most of the main concepts in virology. So
we do have Virology 101 on TWiV, but maybe that's a little advanced. But I must say, Regis, stay tuned because I'm incubating an idea, and I have been for about 6 months, for a very, very basic virology series on a podcast format. So stay tuned. Steven sends us a link to an article. He says, researchers opt to limit uses of open access publications. Another Nature article. Academics are adopting the view that publicly funded research should be available, but data research released yesterday suggests that given the choice, even researchers who publish in open access journals want to place restrictions on how their papers can be reused. Well, I think I remember this another time we talked about putting your data out there and people can't just use it in any old way that they please. We did talk about a similar article a while ago. Well, there will always be problems with any open format, I guess. You know about this, Alan?
Yes. This is a simmering issue that I think is going to be continuing to simmer for a while, because one of the issues with open access is what do you mean by open? Of course, everybody right now is talking about, it should be readable by the general public, but that's only one type of access to a paper.
Yeah.
Can you use a computer to search through and mine data from multiple papers this way? Can you republish portions of it for profit? Can you reprint figures? What are you allowed to do exactly? And there are different varieties of licenses that would permit or restrict various uses of that, and I think most researchers haven't really thought that through.
Yeah, I mean, they're talking here about mining a paper and then republishing aspects of it and that sort of thing, right?
Yeah.
When I think of open access, I just mean letting people read it. I
Right.
think
Yeah.
if we could do that, that would be really good.
Sure.
But
Right.
the next step is harder because then there's
Yeah.
so many different possible ways that I don't know how you could cover them all. Anyway, you folks can check that out. And let's do one more. Alan, can you take the one from Shane?
Sure. Shane writes, "Hi, Vincent et al. I was still catching up on all the back episodes of TWiV and was recently listening to TWiV 130, where you discussed 2 different vaccinia outbreaks. As a little background, I compete in Brazilian Jiu-Jitsu, which is similar to wrestling, and I'm also a referee. You asked the question, why did he wrestle? That made me laugh. People attempt to compete all the time with visible staph infections. I doubt the wrestler in question even cared if he transmitted it to a bunch of people. In the second case, it was specified as a martial arts school, and the source of the infection was not found. It is quite common for mixed martial arts gyms to have people drop in from other schools, or people who are in the area on vacation will stop in and train at any random school. I can see that it would be quite possible that this martial arts school had such a visitor who could have transmitted the vaccine-associated strain to these people. It is not common for schools to do any background check on people to find out whether they've been in the military or had a recent vaccination. Cheers, Shane from Australia."
Yeah, these were the outbreaks where people got a vaccinia or a smallpox vaccine, and then they wrestled and
Right.
they transmitted it to other people.
Yeah, so I guess a dojo is kind of like a gym
Yeah,
where people would be dropping in
Yeah.
and out all the time. And
yeah. I see we only have one more, so let's just read this one. Mark writes, "I am a dedicated follower and fan of your TWiV podcast, and until now haven't felt compelled to mail into the show. I just wanted to say that your recent guest, Dr. Anthony Fauci, was fantastic. He was insightful and interesting, and I thoroughly enjoyed hearing a bit more about the clinical side of virology. I grew up in South Africa and have a special interest in HIV/AIDS and its partner infections, in particular HPV. And Dr. Fauci's insights into the subject were riveting. I really hope you get him on again. I also wanted to say that your virology podcasts have influenced my decision to do an undergraduate degree in virology and immunology at the University of Bristol, England this year. I will, in fact, know if I've been accepted into the course in the next couple of weeks. I look forward to learning more about the subject and will continue to be an avid fan of TWiV." Mark. Good luck with that, Mark. Hope you get in.
yeah,
Yeah.
All right, science picks of the week.
Wait, I just have to ask, does that mean we are totally out of listener email?
Oh, no.
Just what you put on for today.
Hardly.
Okay.
In fact, I think the next week's we will do an all email episode.
Okay.
No. We got so many more. It's
Okay.
great. It's great. Yeah. We ran out on TWiP, and Dickson and I said, "Well, that's the end of TWiP."
That's it. It's over.
And then we got dozens of emails. It was great. It's a good. We have
it was good. It
great
was very
listeners.
good. It was very good.
Kathy, what do you have for a pick this week?
Well, I picked something that a student in our department, Tom, sent to me. I have a couple people I know that listen regularly, and I said, send me some picks. And he sent me this one. It's the Intel Science Talent Search Awards for 2013. And this contest or talent search has been going on for a good long time, I think 72 years or something, but sometime in the recent past, Intel took it over. And they infused a whole lot of money into it. And so they had, let me see, how many students compete initially? 1,712 high school students entered. 300 were announced as semifinalists in January. 40 were chosen as finalists and invited to Washington, D.C. to compete for the top 10 awards. And then this link that I put in is the announcement of the top 10 winners. But there's a picture of the 40 finalists. And they just have such a range of interesting, diverse science that they do. Sarah Volz of Colorado Springs won the top award, and the award is $100,000
Nice.
for her research on algae biofuels. She used artificial selection to establish populations of algae cells. Second place is $75,000. Third place, $50,000. So these are big bucks going into this. Yeah, $1.25 million for 2013.
Yeah.
So I think that's really good. And you should just go and read the 10 different projects they did, and then you can actually go and read the, I think, more about the semifinalists and so on. So it's, really cool to see this many people excited about science and being rewarded for it. Oh, and this was the other thing. They cite, over the past 72 years, these Talent Search alumni have gone on to win 7 Nobel Prizes, 2 Fields Medals, 5 National Medals of Science, 11 MacArthur Foundation Fellowships, and even an Academy Award for Best Actress.
Who's that? Do you know?
No, I meant to search for that, and I got distracted.
Does anyone here know someone, a colleague, who was a winner of this contest? I don't.
Not that I know of.
Yeah, I just wondered if you knew personally. But we're of an age where maybe 72 years. Yeah, I guess there should be some.
Yeah, we're not that old.
Yeah, it would have been the Westinghouse.
Dickson is.
He is that old. Yes,
Yeah.
Was
he's
it?
that old.
This was Westinghouse. What? Okay, that's what I wondered. Also.
Yeah, this was back when I was in high school. This was still the Westinghouse science competition. I forget exactly what the full name was, but the Circle Bar W Ranch was the primary sponsor.
I had a summer high school student one year who was entering this contest, and man, she was amazing. She was just amazing. She was so good in the lab. So these are really talented people.
Yeah.
Hey, Dickson, do you have a pick?
I do. I'm trying to find it right now because I'm still getting used to my iPad, so forgive me. There was a story on Yahoo News today that arose as I was looking at Yahoo News that documented someone who had received an organ from a person who had died of an unspecified disease, and that unspecified disease turned out to be rabies. And this gentleman died a year later after receiving, I believe it was a kidney, from this person who had died in Georgia, as I recall, but it was originally from North Carolina. So he incubated his rabies for 3 to 4 months and then died from it, but no one realized he had rabies, and they've distributed his organs all over the place. And I think this is a very, very interesting thing because rabies usually kills you much faster than that. And here's a year that elapsed before this person actually died from it. And we haven't had that many deaths from rabies anyway because usually you can do something like give the vaccine
this was,
during
in Maryland, is that right?
Yeah, that's right.
First rabies death in Maryland in many, many years.
Exactly. So
that's unbelievable.
I was fascinated by the whole epidemiology of it, so I thought that was my pick of the week.
Wow.
Yeah.
And I'm just pasting in here the primary source. This is from the CDC.
Yes.
And I'm linking to their press release.
Oh, good.
Which will take you, well, as far as you like to go.
Yeah,
Great.
this is all over the news today.
Yeah.
Oh, there it is. Yep, I see that. March 15th, Dickson.
Yeah, so the Ides of March is right. Beware
Alan got
of
the
that.
CDC statement. That's great.
Could he have been saved if they knew that the organ had rabies in it after they had transplanted it into the patient, but immediately afterwards? So they could have given him the oral vaccine. And
not oral, injected.
Still injected?
Yeah.
Okay.
No, they could have saved them. I think they should put so I have talked to transplant ID folks about this, and they don't test for everything when they transplant an organ.
Rabies is
so I think they really should, because it's not hard to do that, right?
Well, how?
But then you're in a hurry. The person dies, you get the organ. Come on, we got to do this.
How can you take an organ from someone with an unspecified cause of death?
Yeah.
Usually it's an automobile accident or some trauma injury, but it's not a death that's questionable.
Didn't they know the guy died of encephalitis?
They said it was the cause of death was unspecified. If he had died from encephalitis, what would be your first thoughts, Vince?
I wouldn't take his brain, his kidney, or anything.
No, but which disease
Well, they
would
didn't
you?
transplant his brain.
Sorry,
They did not.
I know. I would suspect viral encephalitis.
I would too, and it's very common nowadays to have transplantation West Nile virus transmitted. Very common.
Is it really?
Yeah.
So we have to develop really quick ways to assay
right.
for
That's
a lot
right.
of pathogens.
This is very quick. Yeah,
Interesting.
very true, I should say.
Dickson, I'm glad that you are doing productive things with your time. That's a good one. I like that.
Thank you.
Alan, what do you have?
I have a YouTube video that is for anybody out there who is going through the standard hazing ritual for our tribe, which is where you're forced to memorize the Krebs cycle.
Yes.
That may help tremendously to have this. It is a wrap of the Krebs cycle.
Really?
Yes.
You want to play it for us?
And it goes, well, you can hit on you could just click on the YouTube video and it'll autoplay.
Click on it, Dickson.
I just closed up my shop because my battery's running low, but I will do that when I go outside to my main computer.
Yes, so check that out.
A wrap on the Krebs cycle.
Yeah, it's good.
I found that fun, and I think a number of years ago I would have found it very useful.
It's the,
Right.
the periodic table song, right? That's good.
That's different.
Mm-hmm.
It's different. It's a
And
way of remembering it.
the person who submitted the thing to YouTube said that it was a part of their course. They
Yes.
have them write a song.
You're kidding.
Yeah.
Oh, that's great.
Yeah.
That is good.
Why is it different, Dickson?
Well, the other one sung very fast. Very, very fast.
Okay.
Tom Lehrer was a master, but it was very difficult to memorize. And it wasn't in order of the elements either.
Right, and also people aren't generally forced to memorize the periodic table.
No.
Whereas the
my
Krebs
kids-
cycle, this has real application.
Or the theme song from Pirates of Penzance, for that matter.
Well, my son took AP chemistry last year, and he had to memorize the bloody periodic table.
No kidding.
No way.
Yeah.
What a waste of time.
That's crazy.
And now he forgot it by now, right?
Of course.
Of course.
But he did it,
Yeah,
and I helped him.
What you want to do is memorize the principles. Like,
Yeah.
this
Exactly.
column is that, and this column is that.
The
and-
groups,
yeah, but the
groups.
problem is that it's easier to test the memorization of the table.
Yeah, they tested it, and he got it all right,
Yes,
But,
that's what this comes down to. It's what's easy to
but
test.
the orbital information and all that, which you use later on in the course, they look at it because you can't remember all that stuff.
No.
But maybe they're more familiar with the thing.
In every chemistry course I ever took, there was the periodic table was on the wall, so you wouldn't forget it. It was right up there.
Well, I gave him an app for his phone, and it was right there, so he could look at it all day.
How
And
funny.
that really helped a lot.
How very funny.
Now I still have it on my phone.
Wow.
And I just think it's cool to have the periodic table on your phone.
It's very cool.
So you can look at it at any time. Here it is. It's called the EMD periodic table. There are tons of them.
There are.
But it is beautiful, and you can click on any element
Sure.
and see more about
Oh,
it.
by the way, there's a
Great stuff.
Discovery Channel show that's going to air soon called, come on, kids,
The periodic table?
bingo. It's actually called
Wow.
it's called The Elements, and they're going to go through all of them, I think.
What did you say? It was put out by who?
It's on the Discovery
Discovery?
Channel. Yeah, I don't think they're going to do all 92 episodes,
Well,
but
Dickson, when it comes out, maybe you could pick it.
maybe I could.
All right, I have a pick this week which is not science, so I thought I would stray one week because this is something that I use a lot. It is a pen company online, and it is just I have an addiction to pens.
Really.
I really love writing instruments. I always have. In fact, in the day when I wrote grants by hand before computers,
Yes.
to get myself psyched, I would go buy a new pen, and I would really enjoy that.
Great.
And nowadays, I can't buy a new keyboard every time.
No, that would be great.
Well, I did buy a new keyboard this week, which I love, and I'm going to pick that another time.
You did?
Oh, it's so good. Yeah, this is crap, the one I have here.
You're
But anyway,
kidding
that's
me.
another pick, Dickson. This is jetpens.com. They sell Japanese pens who make the best cheap pens in the world, as well as best expensive ones, or among the best. There are all kinds of pens here. They will fulfill your every need, and every month I get an email from them and end up buying pens. My two favorites I've listed here. The first one, and these are inexpensive, but they are technology in pens is so cool now. You could spend a lot of money on a good fountain pen, but these are cheap
could.
things that you don't mind losing, but are still really good. So the Zebra Sarasa 0.4 millimeter pen. This is really fine. I love writing with it. It's really fine. So that's a good one. And the other one is a Signo, which is a 0.38 millimeters, slightly thinner. These are both, and I have one right here, Dickson, so you
I
can
want to
see it.
I want to see
You
it.
can click it.
I verify
It's
the
got
fact
a,
you just handed it to me.
It's got that's the Sarasso. It's got
I'm about
a,
to put it on paper and see for myself.
a rubberized grip.
Oh, this is so wonderful.
And,
Vince, you're not going to get this back. I'm sorry. I want to
It's
keep
only,
it.
it's only a couple bucks. I really feel bad for people who use really cheap pens to write
this
so
is-
much. Like I see my students in my course, some of them have Bics, 29-cent
How do you
Bics.
refill these?
You don't. You just, it's $3. You throw it out.
What do you do with? Wait a minute. What do you do with it after you throw it out?
Actually, you could buy refills.
How do you?
Yeah,
how
they
could you
have
throw
refills
this
for most
away?
pens.
I just said you can buy refills. I made a mistake, Dickson.
You
Okay.
misspoke.
In the presence of an ecologist. I'm sorry.
Well, you're
Yeah,
an
but the pen
ecologist.
The pen is like the Uniball, for example. I'm a Uniball user myself. I totally get what Vincent is saying. My wife is a Zebra pen user. She uses the expensive metal Zebra pens. But the Uniball refill is $1.35. The pen is $1.65.
Right.
So
I just love them. And if you're filling out forms in particular, you can be so precise with these.
And they'll press through carbons if you have to make copies.
Yeah.
And
Really
Well, that was
nice.
going to be my question. If they're so thin, how do you keep them from like slicing through your paper? I think I pressed too hard.
Well, try one out. They're cheap enough.
Use stronger paper.
No, I was totally fascinated. And
no,
one of my
I used to use these Uniballs on carbon forms all the time. They work great.
one of my former students just had a real thing for any kind of office supplies. So when I saw you put this on the show notes, I immediately sent it to her.
Now, this company is wonderful. They just have, I mean, I have bought so much stuff from them. They have all kinds of cool erasers and pencils, and they have really expensive stuff, too. Incredible range, and they're always adding stuff, so it's really cool.
This is awesome.
Just check it out. So it is really science.
Nerds, we are nerds from start to finish.
And before we move off of our own picks, the Intel Science Talent Search winner with who won an Oscar, and the connection to this is most likely Natalie Portman from my Google searching.
Yes.
How cool.
Yeah.
Cool.
Yes.
Yeah, that's great. She's great. So she used to do science, huh?
She's had 2 published papers.
Wow.
Yeah.
Neat.
She's not related to Portman in Indiana by any chance, is she?
I don't know. Her parents were Hirschlags.
No.
Hirschlags. So, in fact, the name on the paper is Natalie Hirschlag.
Did you guys see The Black Swan?
Yes.
Yes.
Fascinating.
It was
Yeah,
great.
that was good.
It
That was
wonderful.
great. She
It
was
wonderful.
so good in it.
Yeah.
Wow.
Yep.
All right, we have a couple of listener picks. Justin, as we already heard, picked the DNA store. And then from Jim, we have a pick, something to listen to on a commute. Massive open online classrooms. So this is, I guess, a discussion about the massively open online educational trend.
Also referred to as MOOCs.
MOOCs. And notice there's no scientists here, but there are people, I guess, who are involved in them, so that might be worth listening to. And they think that the last one I will put up, I have a couple of extras, but we'll do one from Chris Kundayan, who is my buddy over at the American Society for Microbiology. He's the outreach manager. He helped us to make the needle video. He said, I want to send a pic of the day. And this is for people who like RSS feeds, and every one of those would know that Google Reader is going away as of July 1st. And so he suggests using Feedly, which is a very nice reader I have been using for a while now. It's both iOS and browser-based, and it aggregates your feeds in a cool way. So thanks for that, Chris. And that will do it for TWiV 224, which you can find at twiv.tv and over on iTunes. And as usual, if you like us and you're new, give us a rating over on iTunes. Helps us to stay very visible. Send us your questions and comments to TWiV at twiv.tv. Dickson Despommier was once an employee of Columbia University. He's now retired, but he is here at Columbia, New York. Thank you, Dickson.
Pleasure.
For joining us.
Yeah, I had a good time.
Did you?
Yeah, I did.
You really don't care, though.
Still having a good time. Stop it. He says that to me every morning, and I have to rebut that every single day.
I'm really a pain, I know. I'm sorry. But I think you like it. Although today you were very aggressive.
I would not be here if I didn't like it.
I'm not forcing you. You're right.
I'm at the age now that I don't have to do anything that isn't fun
I mean, you don't have guilt?
None.
Wow.
No,
All right.
none.
Well, thank you, Dickson. Kathy Spindler is at the University of Michigan. Thank you, Kathy.
Thanks. This was fun, and I learned a lot.
Great. And Alan Dove is at alandove.com and on Twitter. Thank you, Alan.
Always a pleasure.
I think it's really amusing that at one point you quit Twitter because you didn't think it was useful, and now you're having all these Twitter discussions that end up on TWiV.
Yes.
So
yes. I
did have one comment for Kathy, though. I wanted to know whether Kathy understood the common name for their mascot, the Wolverine.
The Wolverines?
You know what the common name for that animal is? We just found that out recently.
No, I don't think I do.
The bad attitude weasel?
It's called the glutton.
Glutton.
The glut?
The glutton.
Yeah, we did that on Twitter this week, didn't we?
I didn't know that.
The glutton. Go gluttons! Go
because
gluttons!
they get
They eat everything.
giant roundworm, right?
Yeah, they get the kidney worm. Yeah, they do.
Yeah.
But they eat everything. They're just insatiable in terms of their appetite. So I guess maybe that's why the Wolverines are so feared
Okay.
by other football.
Wolverines go viral.
Hey,
Yeah, we had that episode.
You bet. I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.